Welcome everyone to the first edition of the First Coast Innovation Challenge. First time we've ever done this. We're very excited. We have some really great technologies here today. There will be 8 companies presenting. They'll have a maximum of 7 minutes. I will give the presenters a 2-minute warning. And then there will be about 5 minutes for questions and feedback from the panel. Okay, so we're going roughly in alphabetical order. The first company.
Is there a microphone or no mic?
The microphone didn't get set up in time, so
okay.
I think it's a small enough room. Yeah. So allow me to introduce you to Barry Cohen. The company's called Abilis. From the Frederick Innovative Technology Center in Frederick, Maryland.
Good afternoon. I love talking right after lunch because everyone is like really ready to take a nap. So I'm Barry Cohen. From Abilis Life Science, and we've developed a bladder cancer test. So every year, about 1 million people are detected with a condition called hematuria, which is having blood in the urine. And typically, it's might be caught by their primary care physician, but it usually ends up getting a referral to a urology specialist. And the urology specialist, most of the time, will recommend a full diagnostic workup for bladder cancer, which is done usually in an outpatient surgical center, typically costs $4,000 to $6,000, requires sedation, and oftentimes can have complications afterwards, requiring return visits back to a doctor's office or an emergency room. Interestingly, though, of the people that have the condition of the hematuria, only about 3 or 4% actually have bladder cancer. So those diagnostic workups are typically are negative in 96+% of the people getting them. So we've developed a non-invasive bladder cancer test using a standard urine specimen, which can be done from the convenience of a doctor's office, just as people typically do when you go for a physical. You pee in a cup, and what we can do is tell you whether you are at high risk or low risk for bladder cancer. And the idea here is that we pick out the people that are at high risk, and only those that are the high risk group would go on to get the full diagnostic workup. So how would it actually work? So a person goes to their doctor that whether they can see or they can't see that they have blood in the urine, but the doctor has you give a urine specimen, standard way, sends it to a lab where it's analyzed on an instrument, and the output of that instrument is analyzed by our algorithms, and we return back the result to the physician. And right now we anticipate that our algorithms will actually be decoupled from the lab and the instrument itself and be run in a remote subscription-based model. So the accuracy of the test that we have, we're on our first version of the test, it actually was able to detect 97% of the actual cases where the patient had bladder cancer. So it correctly found 97% of the bladder cancers, but equally as important, it correctly identified 78% of the people who didn't have bladder cancer. That's really the big opportunity, is confidently telling people who don't have bladder cancer, you don't, and therefore don't need to go for the workups. The technology driving this test is called metabolomics. Right, so we are able to extract about 2,400 data points in analyzing the urine specimen. And these are not genetic markers, but these are markers that relate to how cancer, the metabolic pathways that cancer has when we analyze the urine. And so what's interesting is because if you think about it, bladder cancer and the bioavailability of these markers, because it's in direct contact with the tumor in the bladder. So it's a really good way of finding bladder cancer. It's a lot of stuff, information here about our pathway, but very simply, we have 3 main milestones we want to achieve. One, we hope to go to the FDA in 2019 with our test and get approval for that. Second, we want inclusion in the AUA, American Urological Association, clinical guidelines. We need the professional support for the use of our test. And thirdly, we need reimbursement from the healthcare payers. So those we hope to achieve over the next 4 years, and our plan is to partner with larger companies, whether they be diagnostic equipment manufacturers, distributors like companies like Abbott, Roche, Siemens, Beckman Coulter, Becton Dickinson, etc., and ultimately partner with the big customers, which would be companies like LabCorp and Quest. So we actually, when we go to commercialize the test, what we will do is allow labs, whether they be hospital labs or big central labs, to run the test in their labs, connect with our software. They would use it. They would only pay for it when each time they run a test using a SaaS subscription model. And we think the actual cost of the test should be somewhere between, we have $600 on here, probably between $500 and $750. And that's because there's an existing category of tests that are used for monitoring people for bladder cancer. It's basically called a urinary biomarker test, and those get reimbursed about $600. So we have an existing reimbursement pathway already in place. And we think the actual volume, testing volume, could be 1 million or more tests per year. Sometimes people have to get tested multiple times per year. The company is made up of the 2 founders, myself and my partner. I'm the business guy and the lead investor. My partner is the scientist who really came up with the test. And we're actually very fortunate to be working with collaborators and advisors from 4 different universities. 2 of them are national KOLs that sit on the guideline committee at the AUA for bladder cancer, writing the bladder cancer guidelines. So they're looking over our shoulders, making sure we're on the right track, and telling us that if what we're doing is going to be good enough to get inclusion in the guidelines. So that's critical. And we are currently in a funding round. We're trying to raise $750,000. I'd like to close $250 by the end of this year. We're going to use that money to set up our lab, procure additional specimens, and we think by closing this round, we will have enough evidence to go to the FDA and achieve that first milestone we hope to achieve. So thank you very much, and I look forward to answering any questions from the group.
What's your first FDA milestone?
Timing-wise?
The one that you want to, you said hope to achieve with the FDA, the first one.
Right, so our product really is a software product.
Yeah.
So really, we're going to go to the FDA with the data that we have. Hopefully, we will have accumulated enough. They may ask us to generate additional data. But that's, yeah, it's going to be with you.
So, don't shoot me, but I have 2 questions. Totally different. One is just IP, because you didn't touch upon that. And 2, penetration rates, because you said how many tests per year, but
Yeah.
what realistically do you think you can do year 1, 2, and 3 penetration wise?
So I'll answer the second question first.
Okay.
Our go-to-market, so what we're going to do is we're going to be basically, once the test is FDA approved, we're going to be offering the test. Until we have a reimbursement, we can run all the tests we want. We're not going to get paid for it. So we will basically make the test available, and we will run tests in our own lab, like a CLIA lab model, but it doesn't have to be CLIA because we're not getting reimbursed, so it's not a commercial lab. But when it comes time to actually going to market, we don't intend to we can't take this test and market nationally. We will have to partner with one of the diagnostic companies that has a sales team in place that can get, I mean, the call points are just way too many.
Yeah.
And I've been previously with a company that tried to launch a test, a small company. You can't do it. You have to partner with someone that has infrastructure in place. And we intend to do that. And your first question was?
Intellectual property.
So we've talked to a number of people about IP. We think at this point we are going to do this as a black box trade secret. We're not going to go and file patents on it. It would just allow people to probably replicate it too easily.
So on that, do you have plans to further increase the specificity and variety of the tests? In terms of false negative, there's always this challenge.
So the key consideration with this test, because everyone currently is being sent for the full diagnostic workup, the implication of a false positive is that they go to get the diagnostic workup. We're trying to reduce the number of diagnostic workups, but not eliminate it.
Yeah, but a false negative.
A false negative is the most important. We have to minimize that. Right now our sensitivity is 97%, which is extremely high, right? But even a 3% false negative rate is still too high. So we're going to tweak it to be as broad as possible to catch all high risk, even though we're catching a lot of false positives.
Based on the 2,000 patients?
The 2,400 data.
Correct. I mean, we are very early, so we have right now about 300 data sets that we're working with right now, and by the time we get to the end of the year, we hope to have that about 600. So, I mean, clearly we never want to miss a true cancer. So we have to make it very loose in terms of inclusion, so every cancer gets caught as high risk.
So do you think about using deep learning to optimize your algorithm?
So the answer is we are using a number of different statistical approaches, whether it's deep learning. I mean, we could spend a lot of time talking about the different methodologies, decision trees, KNNs, and there's lots of different methodologies of doing it. We've started with one method, but we will explore others because there's always different ways to slice and dice the data using different methodologies. So yes. No questions? Okay. Thank you very much.
Thank you, Barry. Very interesting. Good. Okay, next up is the company Avidya. Presenting is Jeff Lynn. They're with BioHealth Innovation in Maryland.
Do you mind if I just
Okay, no,
project?
no problem.
Hi, I'm Jeffrey Lynn with Avidya Technologies. We are a Maryland, Johns Hopkins-based biotechnology company, spun out of the National Institutes of Health in Oxford in 2016. Our focus is on the development of what we call a nanoscaffold technology used for immunotherapeutic applications. Essentially what that is synthetic polymers that we can chemically program to have specific features that allows us to control precisely the pharmacokinetics and pharmacodynamics of therapeutics used in vivo. Now there's a lot of different applications of our technology. However, we focused on personalized cancer vaccines because we view this as the largest possible market opportunity for our technology. We've actually spent the last several years validating this with Bob Seeder's group at the National Institutes of Health, and also done this in collaboration with several other key opinion leaders, including Nina Bhardwaj's lab at Mount Sinai. To date, we've raised about $2.5 million. We've demonstrated superior safety and efficacy in multiple animal species, including primates. And we've validated a GMP process, which we presented to the FDA in June of this year, had the pre-IND meeting, and now have clarity around a pathway to clinic. Based on this, we're raising financing to undertake a first-in-human clinical study with Steve Rosenberg's lab at the National Cancer Institute, and also provide us with a 3-year runway. Now, the impetus for focusing on personalized cancer vaccines is based on the observation that T cells are highly effective for mediating durable tumor regression. For example, most of you are aware that due to the inherent genetic instability of tumors, they produce mutations. Those mutations can be presented as antigens on the surface of the tumor cell. Now, these mutant antigens, or what's referred to as de novo antigens, are sometimes called neoantigens because they look to the immune system as foreign. So a T cell can be primed to recognize these mutations on the tumor cell and mediate tumor killing. However, the tumor can express checkpoint molecules such as PD-1, which blocks T cell activity. Now, most of you are probably aware of checkpoint inhibitors, which have really revolutionized cancer treatment, and they allow these T cells to mediate effective tumor killing, which has led to improvements in survival of patients who were previously refractory to treatments based on chemo, radiation, and surgery. Now, the key to this therapy is that you have to have a pre-existing T cell response that recognizes neoantigens in the tumor in order for immunotherapies to be effective. That serves as the basis to want to develop personalized cancer vaccines, because many patients do not respond to checkpoint inhibitors because they do not have a pre-existing T cell response. Now, the personalized cancer vaccine treatment paradigm is essentially the same for most of the companies in the space. It includes the steps of taking a tumor biopsy, sequencing at the DNA and RNA level, identifying mutations that are expressed as RNA, using bioinformatics to identify which of those mutations will actually lead to an antigen that's expressed on an MHC molecule on the tumor cell. That's a suitable target that we can then include in a vaccine as either a peptide or RNA, including a formulation administered to a patient, and generate a T cell response. Now, there's two different parts of this pipeline. There's the sequencing and identification. This we have partners for. We have focused on actually how do you manufacture this vaccine and how do you deliver it into a patient in a rapid way and have a reliable formulation as a personalized on-demand therapy. Personalized on-demand therapy key aspect is actually critical, because if you consider vaccine companies in the space, such as Neon, which went public recently with a $300 million plus market cap, they're limited by manufacturability and also low immunogenicity for generating T cell responses against neoantigen tumors. Now, I'll just highlight briefly, there's a number of companies in the space that are well financed. They suffer from a major limitation in generating a breadth of T cell responses against tumor neoantigens. They published on this and shown that they only get responses against about 3 to 10% of neoantigens identified from a tumor. And this is especially a problem when we're dealing with lower mutation burden tumors where you don't have many targets to potentially go after the vaccine. You need a much more efficient approach. So the other types of vectors in the vaccine technology in the space are vectored vaccines, which we believe there's an opportunity for partnering here. But I want to focus on these two issues that our technology addresses, which is manufacturability and immunogenicity. The way we do this is we apply a tag sequence to the neoantigen during manufacturing. This allows us to control and standardize the properties of the peptides. This actually vastly simplifies manufacturing, and to improve immunogenicity, this tag sequence allows us to bind the peptide neoantigen to an adjuvant that self-assembles into a nanoparticle that's ideally suited to elicit a high magnitude T cell response against the neoantigens. Now, the keys, as I mentioned, is that we have a great deal of control over the formulations, and importantly, through optimization, we've taken neoantigens that Neon and others with their technology are not able to get responses against, and we can generate high magnitude T cell responses against these. This has actually been demonstrated in a couple thousand animals in three tumor models at the NIH's Vaccine Research Center. The key I want to point out is that if you look at the proportion of responses Neon and others get, they get about 10% of all neoantigens. We're able to get responses against about 50%. That's a much larger breadth of responses. And importantly, by having an improved breadth, we can potentially go into a greater number of tumors, elicit the T cell response that's needed for immunotherapies to work. Just briefly, we've raised $2.5 million to date, done most of pre-IND enabling work, had a meeting with the FDA and expect to be in clinic by early 2019. We're currently raising $12 million for a phase 1 basket trial. We can talk about more of that in the Q&A. We have exclusive licenses to compositions and methods of manufacturing. And we've also been in the press in several high impact publications with our own first author publications in Nature Biotech, as well as ended up recently in Nature Materials being described as a promising therapeutics company. We have 5 sponsored programs in our pipeline portfolio, but what we're raising money for is our lead asset, which is a personalized cancer vaccine. And as I mentioned, we have several partners in the space who are prominent leaders, so I can talk about more, but we have a team of chemists and immunologists who are already in place and working at our facilities, fast forward in Baltimore. To summarize, our technology provides superior manufacturability and induction of neoantigen-specific T cell responses that are critical to the immuno-oncology treatment paradigm and superior to competing approaches. This has been third-party validated now by 5 independent parties, several of which has come to us with term sheets for partnerships. We believe that there's a lot of opportunities in the space. We work with key opinion leaders in immuno-oncology, including Nina Bardwaj at Mount Sinai. Also have opportunities to work with Liz Jaffe at Johns Hopkins. And we'll be doing the first in human study with Steve Rosenberg, who's seen as one of the fathers of cancer immunotherapy. Right now, we're looking for Series A or joint development partnerships with pharma to really advance this and accelerate technology to the clinic. We believe there's a lot of opportunities and happy to discuss those with you. Thank you.
So point of clarification, this manufacturing nanoantigen works with other medicines to make them more effective, or is it standalone?
Right, so the immuno-oncology treatment paradigm is that you need a T-cell response in order for a checkpoint
And
inhibitor
for therapeutic.
or
Yes.
adoptive cell therapy. So the vaccine would be used in combination with checkpoints, allow them to extend.
Right. What did the FDA tell you about getting approvals for combination therapy? Somebody else's drug with your delivery, your novel approach.
Yeah, so for the first in human studies, they're allowing for compassionate use. So off-label use of PD-1 for the patients in the basket trial, but they are certainly amenable to combination. They see that as a path forward. And indeed, most of our preclinical studies have been done in combination with checkpoint. Vaccine alone, just to be clear, is not the path forward for this technology. It's really the combination.
Well, this is maybe a little too deep in the weeds, but this sounds just like similar to Neximmune. Are you familiar with them? Because they're out of Johns Hopkins, and I feel like maybe you split the IP and you got some of it and they got some of it, or what's going on there?
Yes, Neximmune, they are creating artificial antigen-presenting cells. So I think that would be more suitable in a setting where you have leukemia or something, where you're chemodepleted in patients' pre-existing T-cell reservoir or APC reservoir. Here, we're relying on endogenous priming. So we have an injectable vaccine that primes a response in vivo. So we
Okay,
target
so
our-
it's not the same technology.
No.
Okay, it just seems similar in some ways, and it was the same university. Okay, gotcha.
So I have a question about this technology. How large will the combination of your product be? So is that difficult to deliver to the tumor cells?
Oh, so is it possible to actually deliver to the tumor cells?
Yeah.
Yeah, so the way the nanoparticle, so it's 20 to 50 nanometers in size, and the key to the technology is the particle self-assembly. So I didn't get into this, but really the key is that we manufacture a single chemically defined construct with a precise molecular weight and a single peak by HPLC chromatograph. That makes our release testing very simple. So we can go to the FDA and say, this is chemically defined. We induce nanoparticle self-assembly just by addition of PBS solution to the formulation. The nanoparticles we get are the same size every time with any new antigen. That is really the critical features behind the technology. And those 25 and 50 nanometer size particles, if you track them in vivo, if you give them subcutaneously intramuscularly, they go to drain lymph nodes where they prime T-cell responses. Or we actually give the vaccine intravenously, and we can get the vaccine accumulating in splenic dendritic cells, which prime a T-cell response. Those T-cells recirculate and go to the tumor.
Who is your customer, and how do you reach your customer?
So our customer, most likely, unless we build this out, unless you go IPO in the space, you're probably going to license this out to pharma post first in human data. There's precedent for that on multiple fronts. So for example, Turnstone Biologics licensed out based on first in human data in 7 patients.
The question, who will ultimately use your product?
Right,
Cancer.
yeah. So this can,
So who buys it? A hospital, or who pays money for your product?
well, right now, if you look at adoptive cell therapies, it costs about $300,000. We estimate the cost of goods is about $40,000 for this product. On top of checkpoint, that makes it suitable for the cancer patient population. Insurance companies will ultimately reimburse this. If you provide a therapy to, for example, pancreatic tumor patients, which is a patient population that Liz Jaffe wants to advance this technology to, those patients have about a 4-month median survival time before their tumor progresses, even after excising the tumor. If you can show that you can actually improve survival in those patients, we believe there's a possibility for getting reimbursement for this therapy based on precedent.
And is it an all-comers trial? Because I think you have...
Yeah, first in humans, a basket trial.
Yeah.
Non-small lung cancer would be the initial target population. There's about 140,000 patients a year that would be amenable to this treatment. There are also about 30 to 50% of them have responses to checkpoints. Our population within that 140,000 patients would be the ones that are currently non-responsive to checkpoints.
The administration could be separate from the other therapeutics. So your vaccine or activator, along with, let's say, a biosimilar of a PD-L1.
Exactly. Yeah. So the idea here, and I didn't get to expound on this much, and this is partly why. So Steve Rosenberg's group is interested in adoptive cell therapies. What they need though is a pre-existing T cell response, as do many of these other therapies. So our vaccine gives you that pre-existing T cell response that you can then leverage with checkpoint inhibitor or adoptive cell therapy or another mechanism of action.
I have last question about how long for the whole process, because you start with the
I'm
sequencing.
gonna give you the conservative estimate just to not set expectations too. It's about 6 to 12 weeks and $100,000 currently. We know we can get this process down to 2 to 4 weeks and about $40,000, but you have to build out that.
Within that time, the patient may, the tumor may progress.
Yeah, it's possible. Yeah.
But that's why the 2 to 4 weeks is so much better than the 6.
Yeah, certainly. Neoadjuvant setting could be the setting where you would be able to buy a lot of time because those patients have the tumor size, you could come in with the treatment. But the problem there is if you're trying to see quick efficacy endpoints, you're looking at progression-free survival. So that would be the disadvantage there. So.
Okay, I'm sorry, our time is up. Sorry to rush you along. Thanks, Jeff. I'd like to present Srinath Valdapalli.
Valdapalli.
Okay, I pronounced
Thank you very much.
The company is called RiskCall. They're from here in Boston, UMass Boston Venture Development Center.
Yep, incubator.
Would you like to use the mic?
Yep, sure.
And you can do your slides with this.
Thank you. Hi, good afternoon, everybody. Thank you very much for the opportunity to the RISI conference. My name is Srinath. I'm the CEO and founder of RiskCall. At RiskCall, we help hospitals in improving nursing workflow using smartwatches, so efficient and happy nurses can do a better job at the bedside. How we do this? Existing patient-nurse communication technologies are inefficient. Imagine whether it's you or your loved ones in that hospital bed looking for a bathroom or a pain medication help. The wait for an answer to the help using these call buttons seems like an eternal. On the other hand of the call bell, nurses have no idea the reason for your call or the calls from other beds. They walk up to 5 miles per day, creating a physical fatigue, and they bear up to 80 decibels of peak noise, causing alarm fatigue. So the physical fatigue and alarm fatigue is what makes nurses to become insensitive to patient needs, often causing delayed response time, which is identified to be a #1 factor for patient falls and patient satisfaction scores, costing healthcare facilities up to $4 billion through avoidable falls and then cuts in reimbursements. If you look at this industry, this is currently valued at $813 million as of 2018. It's growing at 10%, reaching $1 billion in the next 4 years. The need for integrated communication systems, changes in the regulations are driving this industry. Medicare has already announced reimbursement for nurse call bell system-based quality outcomes. So in response to these changes in the regulations and the antiquated nature of the industry, RiskCall has designed 3 different solutions. To begin with, our market entry product is the RiskCall Business Intelligence Tool, which is a web application software platform which works with the existing call bell system, extracts the data, and provide daily, weekly reports and real-time text notifications about delayed call bell response time. So basically, it's a web application software providing deeper insights on what's happening on the floor between the patient and then staff. Then we have a nursing workflow solution, which is basically add-on on the existing business intelligence tool. This is primarily geared towards the nursing staff to empower them so they can prioritize the calls at the point of care, so efficient nurses can do a better job at the bedside, improving patient safety and experience outcomes. We have our flagship product. Currently, this is not our focus. Our focus is majorly on the nursing workflow solution. But our platform can also be leveraged to provide end-to-end patient-nurse communication using smartwatches. A single tap on the patient device can put the patient in direct contact with the nursing staff as well. So currently we are offering this solution using as a service model. Hardware is leased with $1 a day per device. and software with a 30 cents per bed per day with a $5,000 of installation fee. This is just a market entry price. And with this price point, we see a total untapped market of $1 billion per year recurring revenue in US alone, and total immediately solvable market is $330 million per year. Globally, this is a $10 billion per year total recurring revenue addressable market. As I mentioned, our market entry strategy is to deploy business intelligence tool and nursing workflow solution in 3 to 6 months, and upgrade the system to communication platform in the next 24 months. We have already secured 4 paid contracts, 2 in the hospitals and 2 in nursing homes. We have not deployed yet in the hospitals yet, but we have deployed in the nursing homes. Based on our experience of bringing these customers on board, we see lifetime value of our customer varying from $17,000 to $54,000 per year, based on the service and the number of, based on the service the customers contract to and the number of beds. And currently we are securing these devices at a unit cost of $70, and the average cost of customer acquisition for every successful lead generation is $3,000, with a gross profit margin exceeding 80% in the long run. With this disruptive technology, currently we are competing in 3 different industries. On the bedside, obviously the call-bell system, nursing workflow solution. Currently these are dominated by the smartphones as well, and also the middleware. RISCall, besides addressing these 3 needs using a single platform, hardware or software, we can also leverage the platform for many other applications such as fall detection, vendor detection, and other location-based services as well. And RISCall platform is already 1/3 cheaper than any of the existing competitor in the market. As of today, it's been 4 years we have started RISCall. We raised over $500,000. We generated $100,000 of revenue. We finished the product. We deployed in a nursing home in Pittsburgh, and we have an approved international patent under system and method of utility patents, where we have a proprietary way of achieving nurse call-bell system functionality using wearables. Moving forward, I'm looking for a $1 million of seed fund. With that, I'm going to go to the stage and come back. My deliverables are, that is going to give me a runway for up to 24 months and help me to deploy RISCall 6,000 devices across 64 facilities. And that would also help me to publish case studies, white papers, up to 10, and set the stage for $2 million of a seed fund. And I just want to show you on the financial projection side, this is where we are hitting with the $1 million, and the remaining $2 million of fundraise would help me deploy 10,000 devices across 110 facilities with an annual run rate of $8.7 million per year and $5.8 million of operational profit with a 97% gross profit margin. We have a pretty interdisciplinary team of subject matter experts on the hardware, software, business, and healthcare. We have an experience of executing cutting-edge technologies on a very lean budget. Myself, I work for the companies like Texas Instruments, and my partner work for the health IT as well as medical device companies. Just to summarize, we are an entrepreneurial team. We have a passion on the healthcare, using wearables in the healthcare. We are addressing a $1 billion industry. We are a revenue-generating company, and we have an approved international patent in the US. With this, I would like to wrap up my presentation and open up the floor for questions.
What have you done so far to establish the value proposition?
Sure. We finished the product and deployed in 2 nursing homes in Pittsburgh.
Are you collecting the data though?
Yes, I thought I mentioned here. We reduced 30% of the falls in our first 6 months of deployment. It's been only close to 8 to 9 months, and we still are collecting the data to show the efficacy both on the hospital and nursing home side. On the nursing home side, we are focusing on reducing the falls. Obviously, it helps the nursing homes to reduce the rehospitalization from nursing home to the hospital. Whereas our value proposition on the hospital side is not only reducing the falls, but improving the patient satisfaction scores such as HCAHPS, where it is measured along the light dimensions. Top 4 of them are like how quickly you are getting help for bathroom or a pain medication, and the hospital nurse as well.
Where are you with the production and availability of devices?
So
Or on large scale?
on the large scale, currently I can able to secure the devices at $70, and I have an inventory of...
Not device price, volume.
Yes, I have an inventory of... I can support like 5 to 6 hospitals right now, or healthcare facilities.
Yeah,
But in the long run...
Long run is, how long is long run?
What exactly, I'm trying to
Well,
understand.
let me tell you where I'm headed.
Yeah,
You're onto something here, in my opinion. There's over 5,000 hospitals.
Yeah.
You mentioned nursing homes. There's a huge market in
Yeah.
assisted living or semi-assisted living facilities. I don't know about the IP part, but if you're a first mover and you can really do this right, and you can show those type of economic savings in a hospital, you need to be prepared
yep,
to be in most of the hospitals
yep,
within 5 years.
yep.
That's 5,000 hospitals.
Yes.
X number of beds each.
So 200 beds, there are 1 million beds in US.
Yeah.
Yep. So we did have this discussions with the manufacturer. So right now, there is an expectation from the manufacturer on my side to have a minimum production of 500. So I don't have much control right now, but once I have a clarity on how big, like, depending on the investment size, I'm controlling my inventory, if that is making sense.
So as you think about hospitals, the other issue is sort of integrating into their environments and getting on their networks. And I don't know, have you had any feedback in terms of, what obstacles you might run into there and being able to integrate into those, because they can be really fussy in those environments as well.
So as a communication platform, one of the things which I have taken care from the beginning is to make the IT team happy. So we are not collecting, processing, or storing any personal health information. So in that way, we are HIPAA compliant. There is a second component. Where do they want us to put the device? Is it in the layer 1, layer 2, layer 3? So we always request the IT team to put us on the medical device Wi-Fi zone. So for that, it takes a little bit of a back and forth discussion, but we did have an experience of working with the hospitals in clearing those Wi-Fi level access to our devices.
And the data that you collect,
Just the room numbers, why the patient was calling, and the response time. So it's completely,
but you talk about like nurse workflow and things like that, which I assume they would want to maybe analyze. Where does that get stored? Does that mess with their data on their network, or do you have a cloud
we have a
solution?
cloud, everything is in the cloud, and that's also HIPAA compliant as well. And we are trying to stay away from the EMR for right now.
So are you integrating into Epic or just some other system, not like the big Epics?
We don't integrate with any system. That's what even hospital wants right now. We are a standalone communication platform, just patient to the nurses without dealing with the PHI. So to answer the question, we don't need to unless the hospital requests us saying integrate with the EMR. Until that point of time, we are not doing anything.
Because from my opinion, just having invested in somebody who is, it's a big job.
Yep.
And-
It's another project basically for
Yeah.
me. It's another startup project.
But what, has there been any reticence from the hospitals to?
They're happy actually when I say that I'm not integrating with EMR because these type of systems do not need to be integrated with the EMR right now.
But are they willing to pay the price?
If we integrate,
No, just your
Yes.
price. I mean, so you don't have a hospital yet. It takes a while.
but
to
they
get-
paid us. They already paid us $25,000 for the contract. Of this 100,000, the first 25,000 is from the hospitals. We had a trouble in working with the hospitals. It's getting the contract is easier with the hospitals, but executing the contract is a little bit difficult with the hospitals. So then I moved immediately my focus onto the nursing home because I want to get the data, show the efficacy,
Right.
iterate the product before I go back to the hospitals.
Makes sense.
Okay, time is up. I'm sorry.
Thank you very much, everybody.
That was great.
Thank you very much. That's fine. So I'd like to present Larry Alberts. This is another incubator from University of Massachusetts. It's M2D2 in Lowell, Massachusetts. And the company is called CamMed.
Thank you. Great. So I'd like to ask you to close your eyes for maybe 10 seconds. And imagine that you have diabetes. You have to stick yourself with a needle 5-6 times a day your entire life. Or you have HIV. You have to take a pill cocktail several times a day. Or you have Parkinson's, and you have to take levodopa 3-4 times a day. Now imagine a future where a pump can deliver these medications automatically for you, and it is so affordable that everyone who needs one can have access to one. And it's so unobtrusive that you and everyone else will hardly notice that it's there. That is what we are working toward at CAMMed with our Evo pump. Now, across these and many other conditions, it's been well established that continuous infusion of medications delivered automatically to the pump is superior to people self-injecting or taking pills several times a day. You can more readily optimize dosing, especially when the pump is integrated as part of a closed-loop system, but importantly, people never miss a dose, and this improved compliance contributes to better outcomes. The problem is that the vast majority of people don't wear pumps. The pumps are still far too complicated, far too expensive, too clunky, too burdensome, too visible, presenting such physical and psychological and economic barriers that most people don't use them, and payers only reimburse them for a subset of people who might otherwise benefit from wearing a pump. It is time for pumps to evolve. Every pump that's on the market today is still based on that first, it's the same electromechanical design principle as that first backpack pump, it's hard to see on the left side, that was developed nearly 45 years ago. Now, pumps today are smaller, but they still have many rigid components contributing to their overall bulk and rigidity, and they're really hard to make. They're very expensive to assemble. And they all just have a single reservoir, so they can only deliver one medication. At CAMMed, we have taken a different approach to pump design. Our Evo pump's electrochemical-based design enables us to make it by taking multiple layers of formed, drug-compatible plastics and bonding them together in fully automated fabrication processes. It drives an order of magnitude reduction in cost down to $1 per unit. And it produces a device that is thin and conformable to the body, just like a bandage is. And it has multiple reservoirs, giving it the potential to deliver multiple medications. So there's a huge opportunity to grow a market for pumps that even at these really low rates of adoption today, is $7 billion, to grow that 5 to 10 times or more by stimulating pump adoption, by offering a device that costs less than a pen, yet fully automates delivery, enabling better compliance and control. And it's not just me standing up here saying this, but that influential institutions like JDRF in diabetes see this potential and have partnered with CAMMed to support our development of the Evo pump as an insulin pump. J&J has brought us into their J-Labs portfolio, and we've got a great following across a number of institutions and in other therapeutic areas. So we've gotten great recognition so far. We've won a number of very competitive prizes and grants that, combined together with a seed round that we closed last year, has been funding the completion of 2 iterations of prototypes. The first of which is a stripped-down version of the Evo pump. We completed that in May. And we're now working on a full-scale pump system prototype that we should complete by the end of this year and then demonstrate delivering into porcine tissue by the spring of next year. We've developed our IP independently. We own it outright. We issued our first patent on the core pump engine last year and have been building out the IP portfolio further this year. Now, if there's time, I'll come back and show you in as much detail as you want how the pump works. For now, I just would like to focus on this chart here, trumpet curves. It's an industry standard measure of delivery precision. We did this on our first prototype, delivering Novolog, U100 rapid-acting insulin, in both 1 and 2 microliter doses, and have shown very high precision of delivery. We have a great team working on this at M2D2 with deep expertise in microfluidics, electrochemistry, as well as general device development, supported by a strong set of advisors. Just to highlight a couple from industry. Bill Van Antwerp retired after an illustrious career at a giant firm in pumps, Medtronic Minimed, has joined our team last year and is supporting all aspects of our development. From MIT, Julie, helping us with the electrochemistry and fluidic control within the pump. And medical advisors, Will Shue of Joslin, Ryan Ratz at Dartmouth, ensuring that the clinical relevance of our device is maintained as we continue to iterate the design. And institutional support, as I said, from JDRF and J&J, also the New England Pediatric Device Consortium. And we are just now entering into a strategic partnership with Analog Devices, which is taking responsibility for all the electronics and sensors in the device, key to integrating CGM technology into this. Going forward, we are now raising an additional $5 million, of which we're targeting $3 million to come from equity, and that will enable us to advance the design from one that is delivering into a pig, from one that is then ready for an IDE-ready device after a year's worth of human factor studies and further modifications to the design. So thanks very much, and I'm happy to take your questions.
What's the size of the patch?
Yeah, it's basically about this big. So I mean, I'm sure none of you knew that I was wearing this. It is totally unobtrusive.
Is it flexible?
And it's conformable to the body. I mean, you can't bend it like this, but it's sufficiently conformable to adapt to anywhere on your body except perhaps your pinky finger.
And how long does it stay?
So you'd wear it, all pumps today are indicated for up to 3-day wear, whether it's a patch pump or a durable pump with an infusion set. And the issue there is really the cannulation site and inflammation around it. So initially, we're also designing this for 3-day wear. We would eventually, not in the first gen pump, but eventually like to extend to weekly wear.
What does the flip side look like from we talked about
So-
cannulation?
Yeah, so it's a soft Teflon cannula that is inserted with a steel introducer that pulls the cannula in and then is immediately retracted.
All able to be done by whoever the patient is without tearing themselves up.
Yeah, so the first prototype of the applicator that we developed, this is bigger and clunkier than what it will be, but this is functional. It basically, you take it, you put it over the pump, it lifts up the pump, you place this on your body, you press a button that applies it to the skin, inserts the introducer, and then retracts it in one motion, and then you take this and throw it away. And we make these for about 20 cents.
So diabetes is a cornerstone of this. You mentioned a bunch of other diseases. Is there a particular reason, and are you resting first success on diabetes, or are you thinking about other diseases?
So we are definitely pursuing this as an insulin pump. In part, that is where the biggest market is, and we have funding to do so and anticipate having further funding to do so. It's a very challenging market, and there are other features that we will need to integrate into the pump. I mentioned CGM as well. We are in discussions with pharma companies. There's one has a drug in treating a neurodegenerative disease that we have a term sheet with. We haven't executed the partnership with them yet. As we move forward up to the point of human factor studies, the work on the pump has a 90+% overlap, whether it is for levodopa delivery or whether it's insulin delivery. So we can continue that in parallel up to the point that then we need to start.
So your business model would be to license the technology and make it, but license it to the various drug companies and their various diseases.
That is correct. It is possible, though, that there is a pharma or device company that's a strong participant across multiple therapeutic areas.
That would just be
So kudos on getting JDRF. That's great, and that's a great validator, but also great financially. What are the terms? Do they have a right of first refusal, negotiation, or anything like that?
So I mean, there are. Well, I won't go into the specifics because it's confidential, but it's quite standard for them with their grants to have a royalty arrangement where they recapture the grant plus a small premium.
Okay.
So in that sense, it's not 100% non-dilutive, but it is nearly non-dilutive.
Yeah, know that was your name. That's good.
What's your FDA pathway on this one, in terms of the class of device and
So class 2, 510K, we're looking at Insulet's Omnipod as the predicate device that we would be comparing ours to.
What's the capacity? Are you doing 200U, 300U, or?
Yeah, so initially, I mean, the data that I showed was with a U100 Novolog, where, and again, comparing ourselves to the Omnipod, which has a 2 mil capacity or 200 unit capacity. That's the base design that we're using. So the pump, it's comprised of these multiple reservoirs. So each reservoir is about 1/4 of a mil. And it's very easy to scale up or down the size of the pump, varying the number of reservoirs. And we just make a different mold for the microfluidic connections between them. So there could be a pediatric version of the pump that has, say, 4 reservoirs that's really small. And skin real estate on kids with type 1 diabetes is a big issue. That's one of the reasons why JDR is very interested in this. Or for type 2 patients that require large volumes of insulin, we could have a larger scale device. But what's great about the electrochemical control, we can control the electrolysis process very precisely and thereby control the delivery precision very precisely. So we could have highly concentrated drug formulations in the pump as well, which then enables you to maintain the volume capacity at a low level but still be delivering a large amount of medication.
You can potentially make it smaller. Because that still
Yes.
looks like probably half a miniature with.
Yes.
That's it.
Yeah.
Okay, I'm afraid we're out of time. Thanks very much, then. I'd like to introduce Patrice Milos. She's from Brown University. Would you like to do some more?
No, thank you.
The company's called
Can you hear me?
Metagenomics, and she's going to use her own voice.
I think you can hear me just fine. So thank you again for the opportunity to present today. It's great to share the story of Medley Genomics. So we have come a long way in our quest to cure cancer. When you look at a 125-year tradition from surgery to radiation to targeted therapy, but we still have much more to do to tell really patients that we can cure them. Last year in the United States, 1.7 million people were diagnosed with cancer, and 600,000 people died of cancer in the US alone. And these numbers rise. Only 20% of lung cancer patients diagnosed last year will be alive in 5 years. So it is a disease that affects us, it continues to affect us, and even though we believe we've made great strides, we still have much more to do. It affects all of us. So our vision at Medley Genomics is to provide novel data analytics to individualize cancer treatment, providing hopes of lasting cures for patients. And I wish my colleague who was working on cancer vaccines was still here. So today, what do? We take a slice of a tumor, we profile that tumor molecularly, and we treat the most abundant mutation typically that we find. And these are 4 non-small cell lung cancer patients treated, and what happens? 80% of those will relapse within 5 years. What we do is we use our data analytics to actually describe all the tumors of this all the cells of this very heterogeneous tumor. There are cells that are low frequency percentage that will often result in tumor regression, tumor resistance, and so forth. And so what we aim to do again is categorize all those from the very beginning to diagnose, treat, and actually monitor the disease. So those 4 patients now look like this when you apply our data analytics and show you the different phylogenies, and these are the different cell types that are seen in that single patient tumor using the software that we provide. Would you treat patient 35 the same way you would treat patient 65? Very unlikely based on the molecular mechanisms. So our goal again is no relapse. We fit seamlessly into the workflow of the current day molecular diagnostic laboratory, where we take the data that's generated from sequence and use that in our software to actually define quantitatively the percentages of cells, qualitatively the molecular mechanisms present in those cells, and then use that to create knowledge that can be used by a tumor board for interpretation for patient care. Using this, the goal would be to develop the data from individuals to really build a population-level knowledge that you can look very early on in the course of a patient's therapy to understand what kind of pattern they might see if that tumor continues to develop. So just a few examples of how this is used. So if you are defining the tumor cell subpopulation of one patient here, you have a driver in clone 1 of P53. You describe the number of variants, the percentages of those cells, and then the other subpopulations and their molecular mechanisms, and identify also novel drivers in the other cell subclones. The goal would be to hopefully use this knowledge to think about combination therapy or actually new therapeutic pathways. Another use is for immune response, and we heard the approach of using individualized cancer vaccines where you're sequencing the patient's exome and determining which epitopes may be relevant for a vaccine. And what we suggest is by understanding the clonal structure of that tumor, you can do a better job at choosing those epitopes that you create your vaccine with. And this is just one example where of these 5 different populations of this patient, you can identify specific neoepitopes in individual subclones that would be good for incorporation into your vaccine. So the characterization of tumor heterogeneity I've described talks about the cell phylogeny, the structure and evolution, the somatic mutations, the molecular fingerprint of each tumor cell, the ability to understand the functional consequence, pathways for the discovery, and building the data repository for future understanding of patients. Not only do we do the patient heterogeneity at the individual level, but we can also understand the heterogeneity across populations. And this has been very interesting with a number of pharma partners that we're talking with today. So looking at 1,000 myeloma patients, what is uniquely different about them, applying a variety of algorithms that allow you to answer that question. The MedLead team is comprised of people that are entrepreneurs, they're experts. Ben Raphael, my co-founder, has been the principal author of all the algorithms and data analytics that we used. We were founded out of Brown University, where Ben was. He's now at Princeton. Bear Brady and other colleagues as advisors. And our business success to date, we're about 2 years old, and we're founded in October of 2016. We obtained seed funding. We filed our patents. We've had various challenges that we have been successfully competing in. We have a license agreement for our software with one pharmaceutical company. We were fortunate to participate in the Philips HealthWorks, and probably the most influential accomplishment has been our NCI grant. And I see my colleague here from NCI SBIR, that established sort of the value for many people as to the importance of what we do and so forth. So our customer base right now is focused on early validation studies with pharma and discussions with pharma around the use, and thinking long-term about the clinical application in a SaaS platform manner. We would hope to see revenue in the range of growing annually to about a $60 million business with 4 to 5 years. And our focus right now is in the top 20 oncology pharmas, where we're having great discussions right now and even here today to really do that initial business validation. Our competitive landscape right now is really focused, a competitor of single-cell technology. But I think knowing and having run a clinical lab in the past, the challenges of a single-cell technology for clinical application are extremely challenging, whereas we think using the collective data from a tumor to then deconvolute that mathematically is a better way. We can do other things, and finally, our vision is really to be novel data analytics to understand genomic heterogeneity of complex diseases, offering hope for lasting cures for patients, not just in oncology.
Yes. How do you perform the biopsy?
So we really rely on the physician or the hospital to obtain the biopsy for us. and then the sequencing is performed. We use a partner, New York Genome Center, and that's the data we work with.
So my question is that, so the site you collect the tumor sample will may affect the results for the sequencing results?
Absolutely, and I apologize, I should have been clearer here. Ideally, we're looking for 2 regions of the tumor, or a primary tumor and what might be a lymph node, or a primary tumor and a regressed tumor, and that allows us to reconstruct the phylogeny of that tumor and understand the relatedness. So it's more than one sample because of the heterogeneity that exists in the spatial relationship.
And so, how do you distinguish yourself from a foundation medicine?
Very easily.
Okay.
So Foundation Medicine has a great test with 378 genes that they sequence routinely at a huge scale. We are looking holistically at the tumor and using exome data. So you have an unbiased view of both the tumor heterogeneity, which isn't captured in a single slice of a biopsy. We would like to think of partnering with Foundation to think about how we could add more value to their assay actually themselves. So we are different.
Well, and that gets to the next question, sorry, but like your go-to-market, so what is your business model at this point?
So,
That's okay.
yeah, it is a big question. Right now, the business value is through partnerships with pharma, and the hope would be, as the value is further established, it's a software as a service or a software as a license, or as a collaboration that we could do hand in hand with an academic medical center, with a pharmaceutical company, or we would be happy to be acquired.
Yeah, really?
So the intention is not to produce a report with information that would help the oncologist to then decide whether to treat the patient.
So right now, we feel as though there's a lot of avenues to fit into companies that are doing that. And so we have tended to stay away from then the downstream issuance of a report. However, I think it's a big question. Having run a molecular diagnostic laboratory before, I would think if we need to do that, we would. But it can fit in with a report as issued today by hundreds of laboratories across the country.
So, are you worried about commoditization? So if you're offering a software package, certainly at the moment you may have like a competitive advantage in terms of the software, but it's going to be very difficult to kind of protect that business kind of going forward, which is kind of goes to this question of how do you kind of build this as time progresses and you have other kind of people entering in with new software and stuff?
It's a very good question. I think I spent a lot of time benchmarking the software that we actually licensed, and there really is nothing that addresses it as this does today. We have filed patents on both the software algorithms and then the application of the software algorithms. I didn't even get into the topic of creating the fingerprints from each of these tumor cells to then use that fingerprint for monitoring in blood, because I think we have a very naive view right now of what we do in blood reflecting the primary tumor, and I think we can do a better job of that. So I think what our goal would be is to always stay ahead of where the field of heterogeneity is going, but I think we can be successful, but I'm open to the competition.
Thank you very much, Patrice.
What is the stage of development? What remains to be done?
So right now, the development is the cloud-based platform. So right now, we have the software, all the analytics we can run internally. Right now, we're building it up on the cloud in a HIPAA-secure environment to ensure that if a pharma partner, and this is a part of our NIH or NCI grant, if a pharma partner wants to use the software, they have a secure domain that we can set up for them so that they can use their data without us seeing the data.
Thank you, Patrice.
Yep.
Next, I'd like to introduce Geeta Singh. Geeta is from M2D2 here in Massachusetts, in Lowell. The company is called Mag Optics.
Well, we have a.
Good afternoon. No. So the only thing worse than being blind is having vision but no sight. Imagine being in your teens and you find out that your vision begins to deteriorate so that daily activities such as working on a computer, driving, reading menus becomes very difficult. This is the result of a rare disease called keratoconus. It actually means cone-shaped cornea, and what it leads to is severe vision impairment and often leads to functional blindness. We're Mag Optics, an ophthalmology innovation and development company. Our lead asset is an intracorneal implant. It's used to treat keratoconus and other corneal conditions. We have completed proof of concept in over 20 eyes outside of the US in human feasibility studies. We are based in the US. Our IP is company owned and all in-house. And we are in the national phase with them filed in the US and EU. We operate as a virtual lean structure, and we are now raising capital to advance regulatory. So keratoconus, as I mentioned, is a progressive degenerative disease. It occurs, as I mentioned, it's a cone-like steepening, but it impacts those typically in the ages of 15 to 40, the sweet spot being 15 to 25. Historically, the disease was classified as in 1 in 2,000, but recent studies indicate that number is closer to 1 in 400, which is a 5x increase. And in fact, there are recent studies ongoing that indicate that number may actually increase significantly more. In other parts of the world, that number is closer to 1 in 21. The primary, as I mentioned, as the disease progresses, the vision becomes severely impaired and leads to blindness. Functional blindness. So the standard of care takes two approaches. One is to halt the progression of the disease, and the second is vision correction. In the interest of time, I'm going to speak to vision correction as that's the area that we're addressing. So the typical patient journey is a trial and error. The patient goes through multiple iterations, go through several different types of contact lenses, even go through several combined treatments. The difficulty with contact lenses and some of these other treatments is the fact that with contact lenses, the fit, the intolerance is very difficult for the patient. As well, and with some of the surgical procedures, the outcomes are very unpredictable. This also costs the patient several thousands of dollars, as these are not your standard contact lenses off the shelf. In the last 25 years, there has been little to no innovation in treatments for keratoconus that are patient-specific. So introducing MagArcs, the first patient-centric corneal reshaping platform. It's a radial spoke made out of PMMA material with long-standing FDA regulatory history. It is peripherally implanted, so we are able to elicit the desired structural change in the cornea. The dimensions are standardized, but clinically we use anywhere from 1 to 3 implants to alter the change of the cornea. The secret sauce in personalization is really the number of implants we use and where we locate them. So the combination of the number and the location really allows us to have a very targeted, specific approach. So I mentioned this is a platform. What we have done to demonstrate our platform is we have implanted into 21 patients, 13 keratoconus, 7 presbyopic, and 1 in astigmatism. What we've been able to demonstrate is that not only does it meet a high clinical need in keratoconus, but it also has the ability to address large patient markets like presbyopia and astigmatism, where there is also very limited surgical solutions available. So in terms of milestones, the key takeaway from this slide is from the beginning of the concept of the inlay to our first human implant was 15 months. What we have demonstrated is our ability to develop tech capabilities, de-risk the technology, and all work with a very disciplined, capital-efficient model. So we are now raising $5 million to advance regulatory. What the $5 million will support is preclinicals and independent clinical studies as well as other clinical activities. We will also be, that will allow us about a 2-year runway, and that will then allow us to scale for an FDA path. This is a Class 3 med device, so it does require a PMA. In conjunction with the movement of keratoconus as our beachhead strategy, we will also stagger the development of our 2 other indications, presbyopia and astigmatism, that address much larger patient bases. So in terms of recognition and press, this is just a quick snapshot. We were just recently a JLabs M2D2 company. We were a winner at the Global Med Device Challenge at M2D2. We were also a winner of Proof of Concept and Technology Innovation Awards in Abu Dhabi. And we have several other different accelerators and et cetera. We've also been quoted in several magazines. So the competitive advantage for MagArcs is really unrivaled. As you can see from this, in keratoconus, what exists are a combination of contact lenses as well as a couple of surgical options. In presbyopia, there are 2 corneal inlays and another implantable, which is used to treat presbyopia with loss of near vision. In astigmatism, it has primarily been toric contact lenses. So the real takeaway here is that there is really no single solution that covers the breadth and the scope of indications and provides a patient-specific approach that MagArcs does. So in summary, what we have developed is an unrivaled, proven technology. We're leading with a high clinical need indication, keratoconus, with a plan to stagger the development in the other indications. We have de-risked the technology. We have been capital efficient and milestone driven. But the most important thing is that we have 13 patients now that are living with vision and sight. And coincidentally, our first patient here, Simon, who has now gone on to finish university, and he is now an architect. So we are MagOptics, and we're architects of vision. Thank you.
A few times you mentioned patient-centric, which I might suggest is used in a lot of places and has different meanings and may not be helpful. Going back to your competitive advantage page, I understand that you're more comprehensive, but if we focus just on the left-hand side, tell us, or others tell us, how are you better on the left-hand side versus the left-hand side?
So I'm going to answer that by going back to the standard of care slide, since you asked specifically about keratoconus. So the current path for a keratoconic, so the thing to remember about keratoconus is that it's not only a cone-shaped cornea, but it's an irregular cornea. So the surface of the cornea varies. And if you use things, for the last 25, 30 years, it has been primarily treated with contact lenses, hard contact lenses.
And some surgery.
So, and some surgery, correct. So what the current treatment path has been for a patient has been trial and error because of the ability to fit the cornea. So if you think about having to place a hard contact lens on an irregular cornea. So most patients currently go through either one or mostly a combination of these. So they will try a surgical procedure, then they will use contact lenses. What we've demonstrated in our 13 patients is that our single procedure is able to eliminate the need for many other procedures.
And how long is the lasting? Is this one and done, or does it be refreshing every X number of years? What's the model of use?
So currently this is one and done. The thing with keratoconus is that it is a progressive disease. But again, sticking on the slide, I said there are 2 approaches to treatment. One is to halt the progression of the disease, and that is currently done with cross-linking. What that does is stabilize the cornea, so it becomes a little bit more rigid and structured, and then we're able to implant the inlay on top of that. So what that implies and what that suggests is that the cornea is fairly stable and not changing as much throughout the years or through progression, and currently with our patients, with the 1 or 2 year data, we have not seen any changes.
What's your path to market?
So there are two customers here. The direct customer is the ophthalmologist, and they are the ones actually that would buy the implant. And then there is the second customer, which is the patient. This is a class 3. We obviously have to go through a PMA approval, and our ideal scenario would be to be able to partner with a strategic to help the distribution and get it into the hands of the ophthalmologist. There is a potential, because this falls into a disease and a severely debilitating disease, there is a potential to partner with foundations as well, but the keratoconus foundations, and there are a lot of patient advocacy groups, so there allows some partnering with them as well.
You expect it to be reimbursed by insurance?
So current reimbursement on keratoconic devices and procedures. Cross-linking was FDA approved in 2016. It has been very aggressive in getting reimbursement, and that is currently state by state. I believe they now have 48 insurance companies that have been approved. The devices, meaning contact lenses, have insurance company sort of it's case by case in terms of level of reimbursement. So one of our intentions with the capital raise would be to also begin developing that reimbursement strategy and those networks with reimbursement agents to make sure that we are planning for an aggressive reimbursement strategy with our device as well.
So did you notice some safety issue because you have collected 1 or 2 years follow-up data with those 21 patients?
So safety, I'll address first in terms of the material. So part of our design intention and to de-risk the technology was to use a material that is very familiar and to the eye. So PMMA has a long-standing history in ophthalmic implants and has, so from a safety biocompatibility perspective, we don't anticipate any issues with that. From a safety perspective of the device, the corneal inlay is not a new technology. The concept of corneal inlay is over 50, 60 years old. So in terms of mechanism of action, we have certainly tested that with, prior to human implanting, we tested it in FEA modeling as well as cadaver implants and animals. So we are very confident that this doesn't have any severe safety issues with respect to mechanism of action. It is important to note that corneal inlays are reversible, so this is not like another technology that once it's in, if needed be, we can certainly explant the inlay.
Thank you, Gita. We're out of time.
This is our inlay. I don't think you can actually see it. I've tried to circle it, but you can feel it if you rub your finger over it.
Okay, I'd like to introduce Luis Alvarez. This is another Maryland company from the FITCI incubator in Frederick, Maryland, and the company is called Theradaptive. Would you like to use the mic? Okay.
Well, good afternoon. I know I'm the only thing probably standing between you and coffee right now, so I'll try to keep this over minutes. So Theradaptive, we are developing a platform for targeted delivery of therapeutics. We're an MIT spin-out, DoD funded, and we have developed a platform that allows you to convert any biologic into a ceramic binding variant. I'll show you in a minute why that's important. We are taking a very specific view of what applications to go after first. So we have a very focused program in spine and long bone repair. Long bone repair being funded by the DoD, spine being the largest market that has a well-worn path that is appealing to investors. We have a very interesting setup in that all the molecular development for the lead molecule actually applies to several of the lead indications. So all of the upfront investment that the Department of Defense has made can be applied to other indications without having to develop a new molecule for each indication. We have done some preclinical development that includes GLP IND-enabling studies, and we partnered with some of the top hospitals and surgeons to advise us on how to design our products. So one of the key problems is targeted delivery and retention of growth factors that bring about tissue regeneration. If you can't retain them in a specific volume, you're going to have all sorts of off-target effects. This is true in the spine, long bone repair, following trauma or cancer, and cartilage repair. This is just the orthopedic segment. give you a specific example. Medtronic has a product called Infuse. It's a protein called BMP2. It's put on a sponge and inserted into a screw, which is screwed between two vertebrae that produces bone, forces the two vertebrae to stay together, and that alleviates pain. So they had approval to do the lower back, lumbar spine, and we're telling surgeons, hey, you can use it off-label in cervical, here in the neck. Well, then it got into all sorts of problems because they weren't able to retain BMP2 in that small volume. People who are suffering from suffocation, paralysis, there were many lawsuits. In fact, that cost them dearly, from $800 million in peak sales in 2004 to down to $350 in less than 5 years. And it all boiled down to the problem of retention, targeted delivery. So what do we have? We have a platform, a proprietary application for the development of variations to the protein sequence that turns it into a ceramic binding variant. We hold the worldwide exclusive license for that and have internal IP. We are able to apply this to any biologic and have demonstrated it for a number of other biologics, about 12 different ones. Shown that you can preserve bioactivity and get very tight binding to ceramics. An interesting part of this is you essentially restart the clock on any IP for that composition of matter because it's a new molecular entity. So you can take existing molecules that are off patent, existing ones that are on the market, and restart the clock. So it's very appealing for acquiring partners or partners that maybe want a license. So what does it look like? Here's an example. You may have a ceramic implant for bone void filling. It's a one-step dip and treat, and now it becomes coated and bioactive. So the entire surface area is coated with that biologic, and any cells that migrate over that area will experience a stimulation of that biologic. In our case, we've chosen to lead with bone morphogenetic protein 2, BMP2, which forms bone wherever you put it in the body. So we can put it on sintered ceramics, putties, injectable ceramics, ceramic granules, and now, most interestingly, the 3D printed ceramics, which are making strong inroads into orthopedics. But beyond orthopedics, you can also get into targeted delivery of therapeutics to soft tissues or for localized oncotherapies. Those are not areas that we're pursuing, but ones that we make known to others in case they're interested in doing a deal with us. So what's the net effect of that? You get increased biopotency because you don't have diffusion away of the biologic. You can get away with lower doses. And you also don't get a spike release like you do with a lot of the biologics. So that addresses the safety concerns while retaining efficacy at levels that are higher on a gram-per-gram basis. So what are the lead markets? Like I said, spinal, long bone repair, cartilage repair, and CMF dental. The interesting thing is that the same molecule that you develop for one can be applied to the others. So Medtronic, for example, has a single BMP2 product with indications in spinal, long bone repair, and dental. So there's already a precedent for this. The only thing that changes is slightly the design of the ceramic carrier, which is very inexpensive. So what's the market now for BMP2 alone? It's recovered back to about $610 million a year in sales in 2016. So there's a well-established, mature market that is slowly creeping back. Many physicians are a bit gun-shy still to jump into the BMP2 world, but there is appetite. They do see it as the gold standard to regenerate bone and to do spinal fusions. So we see, by us addressing this safety concern of retention, we address one of the key limiters to market adoption. Okay, so long bone repair, we unintentionally, I guess intentionally, hurt some rats, but in a good way. We recovered them back to health because what we were able to do is fill in a very challenging bone defect gap and get 100% union at 8 weeks in a study where we compared ourselves head-to-head against Medtronic. So this is a GLP, it's called GLP IND enabling study we performed in Australia, and we were superior to Medtronic on all metrics, including ultimate load to failure, where you break the bone again after the last endpoint to show that you have stronger bone formation than the competitor. So this has been met with very good enthusiasm across the industry because there are many companies that make ceramic products for bone filling, but they don't have biologics. So we were approached by Ortho Rebirth in Japan, and now they have generated some revenue for us. We're partnered with them. We're doing a study at the Cleveland Clinic showing goats to repair a very challenging defect where we place our biologic on their bone void filler. So it's an interesting model that allows us to generate revenue before having FDA approval. Also, we're doing a spinal program. This is what we're raising VC funds for, and essentially the same product development path that Medtronic followed. So this is a Class 3 PMA path combination product. We also received a DoD SBIR for cartilage repair, so we have a phase 2 going on now. And we made our system interoperable with the Arthrex system as much as possible. We want to be a platform that others can use with existing hardware. The dynamic in orthopedics is if you make your product interoperable with the hardware that already exists, adoption is much quicker. This is our pipeline, DoD funded in trauma, long bone repair, and we're looking to accelerate mostly in spine by raising our funds. Yeah. Our team, John Greenbaum, the regulatory advisor, 124 product approvals under his belt, VP at St. Jude, and then Yi Arnold, who we took away from Osiris Therapeutics a few months ago. I'll put a plug in. We are looking for a COO. Industry leader. Okay. Our board, I'll just mention Mike Azemski, who chaired the FDA panel that approved Medtronic's Infuse, has been a wealth of knowledge for us. We're in Frederick. I don't see the FTSE representative here, but I'll put a plug in for them. And what we're looking to do. One thing I'll also mention here is we generate efficacy with the first patient in a phase 1 because it's bone formation. So you can see efficacy from day 1. You're not waiting. The second thing is that you have near zero efficacy risk because we know BNP2 already works. It's an improved product. So this is, in a sense, you might consider this a biobetter. And I'll leave it there. I'll take any questions from the panel.
What is the longest safety or tox data that you have? And thinking about the FDA strategy, we know that ortho division is notoriously difficult in a way. Oftentimes they will come back to you and say, how about 3 years safety data, how about 5 years safety data? Just thinking about that, right? And what's the plan forward? How do you take the product through the end process?
That's a very good point. I think one of the things we're doing to preempt that is we filed an RFD to get a designation very similar to Medtronic's Class 3 device designation, and show that we haven't modified any of the natural amino acid sequence for the human part of BNP2, so we shouldn't have any antigenicity. But we're prepared for the eventual, maybe bad news that people didn't ask us for a long study. There's nothing we can do until we get to that point.
So what's the IP?
We own the IP. We have a license, worldwide exclusive license from MIT, and we filed additional patents to expand the coverage.
Are there any restrictions with the DoD funding?
None. So the Big Bill Act, all IP reverts in cases where the government wants to use it. Yeah. Only national emergency.
There's a lot of companies that are doing this, the typical regen med companies. How do you think you're stacking up against some of those?
I think a lot of the companies that purport to do regen med are saying we have a cell, we have a biologic, put it in there. And they're not telling you how they're going to keep it in there. And they don't have any strong evidence showing that the effects are lasting in that context. Many cell therapy companies suffer from this. And there are only very few playing in the biologic space. So Wright Medical, Stryker, Bioventus. And in fact, Bioventus and Stryker just divested their assets. And so there's a big move now in orthopedic biologics to consolidate. I think it's very timely.
I noticed on one of your slides you had snuck in there soft tissue. Kind of curious about sort of wound healing plays and have you is that sort of a natural extension of this?
We have thought of many ways in which you could use it. For example, a sub-wound therapeutic delivery, which is different than a topical application. And you can compose the ceramic particles to be smaller than the size of the cell, so it's not irritating. So it's a very interesting space, but we've focused.
That sounds so great.
Okay, thank you, Luis. I'm afraid we're out of time. We're running late, and we need to get on to the last presentation. Thanks very much. So, Dan Mazzucco from Pennsylvania, from UPenn. The company is ZSX Medical.
Thanks. Again, my name is Dan Mazzucco. My company is ZSX Medical. And I'm here today to invite you to join with us as we bring zip stitch clips to market. If you want to make a difference with a surgical product, you need to find a big problem in a big procedure. Our big procedure is laparoscopic hysterectomy. Hysterectomy is the surgical removal of a woman's uterus. It's one of the most common surgeries we do in the United States. In fact, 1 in 3 women in the United States will have a hysterectomy during their lifetime. We used to do hysterectomy by large incision, so imagine a C-section scar. But over the last 15 years, there's been a gradual trend towards minimally invasive or laparoscopic surgery, so that today, laparoscopic hysterectomy is a big procedure. It's not enough to have a big procedure, though. You need to have a big problem in the big procedure. Our big problem is closing the cuff. So the uterus is the size and shape of the fist. When we remove it, the tissue remaining is called the cuff because it's about the size and shape of the surface of the cuff. And as I've talked to more than 100 OBGYNs, I hear over and over the same refrain. Closing the cuff is the hardest part of surgery. 90% of gynecologists can't tie knots laparoscopically. It's really difficult to do. It's like tying your shoelaces with chopsticks. So what does that lead to? Well, it leads to 2 things. One is it takes a long time. It takes 15 to 30 minutes to close with sutures compared to 4 minutes with zip stitch. Another thing that is a consequence is poor healing. And the worst thing about poor healing is dehiscence. So dehiscence is when you close the wound, it doesn't heal, it opens back up. Now you've got a woman coming to the emergency room. She's likely going to need a repeat surgery. It's a horrible disaster. How common is that disaster? It used to be exceptionally rare. When we did open surgery and it wasn't difficult to throw loops and tie knots, it was about 1 in 1,000. But once we moved to laparoscopic, now it's almost 50 times more common. It's about a 1 in 20 event. So this is a big problem. And it tells us big problem, big procedure, this is worth solving. What's our solution? Our solution is zip stitch clips. It's a series of absorbable clips and a gun to apply those clips. And I'll just show you how this works. This is actually in a device called a box trainer, which is used to teach surgeons how to suture laparoscopically. You just bring the tissue together and bang, 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 you apply the clips. The idea is it is fast, 4 minutes instead of 15 to 30 minutes. We're taking the hardest part of surgery, we're making it easy. We spread the load out over a region. You don't get these high stresses with sutures. And there's no needle, so there's no needle sticks hazard. The clips remain in place and degrade over the course of time in surgery, after surgery. Who's developing this product? I'm a biomedical engineer by training. I have a doctorate right down the street from Harvard Medical School and MIT. Prior to my time in ZSX Medical, I was in charge of developing a surgical product, which we brought from first-time manufacturer through phase 3 clinical trials. That product is now helping people, so I've been through this before. I'm not going to go through my whole team except to say that in each of the key areas of development, we have top experts in place. In addition to our in-house team, we like to rely a lot on the GYN community. So I've personally met with more than 100 OBGYNs, first to get their feedback on product needs, and then as we develop the product to get their feedback on prototypes. From that group, we have selected a small subset of key opinion leaders with whom we have deeper relationships. And then finally, the work we're doing has buy-in from some of the best minds in the nation, including the National Science Foundation, who has supported us to the tune of over $1 million. Now, I want to show you a little bit about our data. I don't have time to get into all the data, but just 2 specific studies I'm going to show. The first is in the sidewall in the flank, where we made, this is an animal model, we made an incision like a cuff, and then we put either sutures or clips on, and we let it heal for 3 weeks. And this is what we saw. So if we don't make any incision, our baseline strength is at this level. If we make an incision, put a suture on there, after 3 weeks, it's healing, but it's not really quite back to where it was because we made this incision and it's still healing. Let's contrast that with zip stitch. Oh, it didn't show up. That's bad. So we'll contrast it with zip stitch. It's going to show up right here. It's actually right back with the uncut tissue, and I can show you afterwards if that slide didn't show up. It's actually statistically significantly better than sutures. So we got better, faster healing compared with sutures. I think it's on the next slide here. Let's see. Nope. Okay, well, so I'm going to jump to the other data real quick, which is going to be after the end here. Here. There's your data. This is actually somewhat exciting. What was actually much more exciting was when we actually put it in the hands of surgeons. So we had surgeons using sutures versus using zip stitch. And these are surgeons that have used sutures for 2 to 30 years who've used zip stitch for 2 to 30 minutes. After just to close one cuff, it took 30 minutes for sutures, took 4 minutes for zip stitch clips. So that's really an absurd reduction in surgical time. These were the data that got the NSF excited enough to give us $1 million to go forward. So now I'm going to go back to where we actually are going. So I'm not going to go through a whole history. I'm just going to start with when we got financing for our first in human studies. That was early last year. Great. We did a first in human study, 10 patient usability study. In the meantime, we were doing a a large animal efficacy study. We completed both those studies, followed the patients for 1 year. Everyone did well. It's very exciting. And then we've spent the last 5 months talking to FDA about that, and now we have clarity. That brings us to today, and that's what I'm inviting you to participate in, which is our current raise. And I can talk to you about the details of that raise after or in the Q&A. But what we're going to do with that money is what I'm going to tell you now. We have to do a 30-patient safety study, FDA mandated, which will lead to 510K clearance and initial sales. So our raise now is for that study, the 510K, and the initial sales. Now, the last thing I want to leave you with is, yes, we're excited about hysterectomy. We think hysterectomy is a great opportunity, a real need, but we see opportunities all over in women's health for soft tissue closure inside the body. So just in women's health alone, almost 5 billion procedures a year, $2 billion of opportunity for women's health. So we're focused on hysterectomy today, but downstream we see opportunities elsewhere. So I know I said a lot of things, I said it really quickly. If you only get 2 things out of our presentation, we have a product that surgeons love, and we have a very clear path to 510K clearance. Thanks for your time.
What is the predicate device?
Predicate device is a product called LaproClip, which is a Medtronic device now. It's used for cystic duct closure. Our comparator device for all of our studies is sutures. And you may have noticed it said 4th FDA pre-sub. That choice of predicate and choice of comparator is all based on FDA communications.
And what is the FDA looking for in the safety study?
I think they just want a clear safety signal. Like basically they said that's more than 10 patients isn't enough. We want to see 30 patients in the study that is designed to show safety. That study was really looking specifically at usability, how easy is it to learn. They want to follow patients for 6 months with a safety protocol.
And what have you raised? How much, and what are you raising, and what have you raised to date?
Sure. So we are raising up to $2 million for this next stage. We have an open convertible note, which has, as of today, is like 400 and something thousand, or maybe just under 400,000. But we're actually looking for someone to lead that up to $2 million round.
You see application in any other surgeries?
Yeah. So actually, our patents are around specific things in our design that we feel are very translatable to other areas. And so we've actually identified, I know I'm showing a lot of things here, but we've identified a couple areas where we would say, yeah, we can do this. So cesarean delivery, huge market opportunity, like many procedures, and huge need, right? You close it once a C, always a C. Why is that? Well, the uterus doesn't heal as well as it could. Knee and hip replacement, that's fascia closure takes a long period of time, so orthopedic. And then any abdominal procedures where you've got fascia closure, we would see opportunity.
Go to market.
So, not going to dump this on the market. We're going to start with a few hospitals, so like 3 hospitals, 6 surgeons for the first 6 months, generating clear data. And this is all the hospital makes the purchase, so we need buy-in from physicians because they choose it and use it, buy-in from the hospitals. We have a pretty good value proposition in the hospitals because just without any downstream data, we save them a lot of time. Right? And then downstream you can show reduction in complications, hopefully, after you've done enough subjects, enough patients.
Mm-hmm. What do you actually make the clips from, and what's the resorption time?
Yep. Great question. They're made of polydioxanone, which is the same as PDS suture. In molded form, they've been used in the body for 20 years, so it's a great safety history. Unfortunately, we had to repeat every one of those tests anyway, but at least we...
FDA wouldn't...
No, they wouldn't take it, but that's okay. We knew we would make good results, and we did. So it resorbs over the course of 6 months. It just needs to hold for 2 weeks, and then it dissolves the rest of the time.
Is the only real value the reduction in time required?
No. There's a lot of value, but to the physician, the real value is that it's really like, imagine tying your shoelaces for 15 to 30 minutes. It's a really sucky part of the procedure, and we make it easy. In fact, it's probably the driving force behind laparoscopic hysterectomies going robotic. So for surgeons, this is a game changer, at least for gynecologic surgeons who are doing routine hysterectomies, it's a game changer. For the hospitals, there's a couple different value propositions, but one is certainly, an immediate one would be time savings. Sure.
Thank you, Dan.
All right, thanks.
Well, I would like to congratulate all the presenters. I think they were fantastic. I was very impressed. And thank you to our panelists for the great questions and feedback. And we very much appreciate it. Have a great day.
Thanks.